DNA. Well, actually, not really. RNA. RNA, we haven't talked too much about. We looked at the sugar, which distinguishes RNA from DNA. We also talked about the nitrogenous base, uracil as opposed to thymine. But we didn't talk about the secondary structure of RNA. One thing that I mentioned early on was I mentioned that ribonucleic acid is really good at hydrogen bonding. I think that it's pretty common if you talk about RNA to just say like, oh, RNA is a single-stranded molecule, whereas DNA is a double-stranded molecule. And I think that's pretty fair. Um, but that's kind of half the story. RNA plays some major roles in the cell. For example, although it's commonly single-stranded, it can form these really crazy structures. It folds into a number of those different structures, and we'll look at those in just a second. Now, in terms of what RNA is, or yeah, to sum up RNA, you could say, well, it's mRNA, it's rRNA, and it's tRNA. mRNA is messenger RNA. It's the messenger that goes from the DNA to the ribosome to make a protein. So that's kind of, it's sometimes common to refer to a mRNA transcript. That's the conversion of a DNA molecule to what's going to be coded for a protein. rRNA, the ribosomal RNA, has a structural and functional component, or it is the structural and functional component of the ribosome. Okay. The last of your major RNA structures is tRNA. That's the molecule that carries amino acids to convert an mRNA to a protein. tRNA, also known as transfer RNA, is basically it's almost like a delivery truck bringing an amino acid to the ribosome so that you can translate a protein. Now, RNA structures, I mentioned this earlier, folds into numerous complex structures. Okay, cool. Let's see what you got. And actually, before I go on to that, what I'd like to say is that single-stranded RNA, like mRNA, for instance, typically exists as a single-stranded molecule. It's very unstable. And the reason that it's very unstable is because it predominantly exists as a single strand. That single strand can hydrogen bond with things, can hydrogen bond with water. It can have a dipole ion interaction with sodium. It can hydrogen bond with an amino acid. And my point there is that if a single stranded mRNA molecule hydrogen bonds with a molecule way over there, and then with an amino acid over there, they're kind of pulling in two different directions, in which case this molecule might be basically ripped in half. Now let's talk about these RNA structures, though. So RNA structures, you got that single strand. Single strand transcript. Yeah, here, mRNA. Smiley face. It's susceptible to hydrogen bonding, so that makes it a little unstable. And yeah, it can hydrogen bond with the environment, but if you had like a G here and a C here, those could hydrogen bond with one another, right? Yeah. Um, and you see that sometimes. Single-stranded nucleic acids, that what they do is they kind of wrap up on themselves. And when they do that, you form these really kind of neat structures. You got a hairpin loop over here. You can form a number of different and unique helices. Then there can be parts where, hey, these all like A and U, this hydrogen bonds really well. A and A, little off. And then, but these right here, these all hydrogen bond really well. And maybe there's sort of some like offset where they don't hydrogen bond perfectly, which could produce either some sort of little bulge in that molecule or a little loop in that molecule. And in this figure right here, that's kind of what I'm trying to illustrate. We've got some hydrogen bonding going on here, and then, oh man, we got some chaos going on there where there's not necessarily hydrogen bonding. Okay, so let me just say it can kind of get out of hand. And man, buckle up. That's that's nerve-wracking. But those unique structures formed by a single strand where there's a bunch of hydrogen bonding, internal hydrogen bonding, gives rise to a structure like this. This is a tRNA. Okay, and we've got our amino acid attachment site. So our amino acid gets basically covalently bound there. We got a bunch of hydrogen bonds. We got a couple of loops. This is the region right here, what's known as the anticodon, that matches up with the mRNA at the ribosome. And so, hey, man, yeah, I would say like it's a little bit complicated, and there's some crazy structures. That's what happens when a molecule can hydrogen bond with itself. And ooh, wow, it's a lot going on. But it can't get more complicated than that. And man, the number of times that biologists and biochemists have used the phrase, it can't get more complicated than that. The number of times they've used that phrase and they've been right, I think it's still pretty much zero because there is an example of a ribosomal RNA. This is the small subunit of our RNA. And this is so complicated that it's tough to even know where to begin. I see a bunch of numbers, I see a bunch of circles. You got to zoom in a little bit to see, oh, yowza. This right here, where there's that 620, that would be an example of a hairpin loop. Between the 620 and the 630 and 610, well, that's 630 to 610. That's where we got one of those internal loops. What I see here is that there are tons of different structures that have emerged from a ribosomal RNA. These rRNAs are pretty fundamental to translation of proteins. Oh, because these are the things that basically help a protein get assembled. All right. That what I wanted you to see here was just kind of the complexity and diversity of our RNA molecules. What are they? The three classes of RNAs in a cell, and how they have varied structural complexity and basically what they look like. So I hope this was helpful, and I hope you have a good one.